everyone, I'm Shahnas, MSc student from University of Calgary. Today I'm going to present my thesis project on pathogenesis and host responses of Canadian 491 infectious bronchitis virus infection in chickens. Infectious bronchitis is an economically important poultry disease caused by infectious bronchitis virus. There are many types of infectious bronchitis virus exist in the world, and this IBV 491 is one of the prominent IBVs, first reported from Europe in mid-1980s. And it quickly spread all around the world except Australia and USA, and also known to cause infection in multiple organs like gastrointestinal tract infection, urinary tract infection, pectoral myopathy, and also they also can infect the immune organs causing immune suppression. And this Canadian IBF 491 was first introduced into Canada in mid-2011, and the isolation rate was very high during the period of 2012 to 2014, and these affected flocks were also presented with respiratory tract problems and production problems. As there were no research studies on this Canadian IBF 491, we wanted to investigate the pathogenesis, its tissue tropism, and host responses. So our research questions were, is Canadian 491 IBV oscillate capable of causing defects in reproductive tract in layers, leading to reduced egg production and quality? And is this Canadian 491 a nephropathogenic strain and induce host responses in the kidney? Let's move on to the experimental design. We conducted two experiments for the analysis of pathogenesis and host responses of this Canadian 491 IBV in chickens. The first experiment was in laying hens, and the second experiment was in young birds. So the first experiment was to investigate the impact of Canadian 491 IBV on egg production, egg quality, and also its effect on kidney in laying hens. We used 28 weeks of age laying hens for the experiment. 12 birds were infected with intratrachea and ocular route, maintaining 12 as control. The birds were kept for 10 days and daily monitored for clinical signs, egg production, and egg quality. Regarding egg quality, we monitored the internal and external quality of the eggs, especially the albumin thickness, any structural abnormalities, the egg weight, the height and width of the egg. And at 3, 6, and 10 dpi, swab samples were collected to monitor the IBV excretion via oropharyngeal and cloaca route. And at 6 and 10 dpi, 6 birds from each group were euthanized to collect blood and tissue samples. Now let's move on to the results. Based on the egg production and egg quality data, we didn't see any drop in egg production compared to control and any changes in the egg quality. Therefore, we can say this Canadian 491 IBV has no impact on egg production or quality. Next, we also investigated respiratory tract, reproductive tract, kidney tissues, proventriculus, and cecal tonsils for IBV genome load. And we found the presence of IBV genome at 6 and 10 dpi in all the tissues, indicating this Canadian 491 IBV has a very broad tissue tropism and capable of infecting multiple organs. The serum samples were evaluated for the presence of anti-IBV antibody. At 6 dpi, only 2 infected birds showed positive results, but at 10 dpi, all the infected birds were positive for anti-IBV antibody. and at both time points the control birds remain negative. To evaluate the nephropathogenicity of this Canadian 491 infectious bronchitis virus in adult laying hens, we did the histopathological analysis and immune cell recruitment analysis in kidney tissues. So we noted IBV-induced changes in infected kidneys, and there were no significant results in control birds. The image A shows increase in mononuclear cell infiltration, and the image B shows the dilation of kidney tubules. And in image C, you can see the epithelial cell necrosis, and image D also represents the increase in immune cell recruitment. As I said, we did the immune cell recruitment analysis using immunofluorescence technique, and we analyzed for CD4 T cells, CD8 T cells, and also macrophages. The image here shows the increased recruitment of CD8 T cells. The yellow color represents the CD8 T cells, and blue color represents the epithelial cell nucleus. And we have noted increase in the recruitment of CD4, CD8, and macrophages at 6 and 10 dpi in kidney tissues compared to control birds. The second experiment was to investigate the nephropathogenicity in young chickens. For this experiment, we used 1 week of age chickens, and 15 chickens were infected with IBV 491 via intratrachea and ocular route, maintaining 15 as control. The birds were kept for 10 days, and during that period, the birds were monitored for clinical signs. And at 3, 7, and 10 dpi, 5 birds from each group were euthanized to collect blood, tissues, and also swab samples. When we consider about the results of this IBV 491 in young bird experiment, we monitored the body weights of these infected and control birds at 3, 7, and 10 dpi. And we have noted a poor weight gain in infected birds compared to controls, and the difference was significant at 10 dpi. We also evaluated the lung and kidney tissues for the presence of IBV antigen by immunofluorescence technique, and we have noted the presence of virus in those tissues from 3 dpi onwards until 10 dpi. We also investigated trachea, lung, and kidney tissues for histopathological changes, and we noted IBV-induced histopathological lesions in infected birds, and there were no significant lesions in control birds. So at 3 dpi, mostly degenerative changes were seen, and at 10 dpi, increase in mononuclear cell infiltration and also regenerative changes were seen. So here these images indicating the deciliation and also hyperplasia of lymphoid foci and also increased immune cell recruitment in kidney tissues and hemorrhages. We also analyzed kidney and lung tissues for increased immune cell recruitment. We monitored the recruitment of CD4, CD8 macrophage cells, and this image indicating the CD8 cell recruitment. The yellow color shows the CD8 cells and blue color shows the nucleus of epithelial cells. So we have noted increased recruitment of CD8 and macrophage cells from 3 dpi onwards, but CD4 cells were only significant at 10 dpi. So based on the results of these experiments, we can come to the conclusion that this Canadian IBV 491 isolate has a very broad tissue tropism, but it does not cause egg production or egg quality problems. The variant is nephropathogenic in adult and young chickens and capable of inducing host responses. I would like to acknowledge my supervisor, Dr. Faisal Karim, and all my supervisory committee members, the lab members, and the funding agencies, Canadian Poultry Research Council, Egg Farmers of Canada, Agriculture and Agri-Food Canada, and University of Calgary. Thank you for giving me this opportunity, and I'm happy to take any questions.
Thank you so much for that. We do have a question in the Q&A box, and I would remind attendees to feel free to type your questions into that box and we'll forward them to the speakers. The question is, do you think the age of the infection of the hen will matter for the effect on egg production?
Yeah, because the effect on the egg production depends on the age as well as the IBV variant. So there is a syndrome called false layer syndrome that especially happens when the infection occurs during young age, especially during first weeks of life. So if the infection occurs during young age with certain IBVs like QX or with certain IBVs, then they can cause false layer syndrome. So those chickens will not produce eggs throughout the life. So we checked whether they can cause any drop in egg production during the laying period, but we didn't observe any defects in the egg production or quality. Hope I answered the questions.
All right, we do have another question. Have you checked the presence of the virus in eggs, and could it be a human safety issue? So a human safety issue.
No, we didn't check the presence of virus in the eggs, but we checked in the reproductive tract and we found the presence of IBV genome in the reproductive tract, but we didn't check the eggs.
Okay. We still have a couple of minutes and I will ask a question. So my understanding is IBV is a coronavirus, and I'm just wondering if you've seen any parallels with what's been going on with the global pandemic compared to your work in IBV in chickens, or is it something potentially interesting to bring up when you talk with friends and family, for example?
Yeah, these coronaviruses, the mutation is very high in coronavirus, especially this infectious bronchitis virus. The mutation rate is very high. That's why the vaccine failures occurs. But the COVID-19, I think, still the vaccine. In infectious bronchitis virus, there are a lot of strains, different genotype. So it's very difficult to control with the available vaccines. But in COVID-19, I think still the available strains are response to the vaccine. So the mutation rate is comparatively low in this COVID-19.
Yeah. I appreciate that.